So I'm just starting recording now. Welcome everyone to the Experimental Medicine Graduate Student Society, the Graduate Association of Physiology, and the Graduate Association of Pharmacology and Therapeutic Students monthly seminar on experimental methods. As you may well know, our award-winning series has been going for several years now, and we've continued to share the specifics and the implications of the techniques used by our trainees. So if you'd ever wish to present a technique that you use in the lab, you can find my contact information at the EMGSS website, emgss.org. So before we get into our presentation today, I want to invite everyone to next month's seminar presented by Benjamin Lebeau of the Witcher Lab on high-C and other chromatin conformation analysis techniques that have revolutionized the field of epigenetics. So this will be held on November 25th at noon. And I'll share a link of it afterwards, but you'll see links on listservs to come. So for the presentation today, I'd like to welcome Dr. Guillaume Butler-Laporte, a clinical trainee in the Brent Richards lab, who's currently working on the biobanks québécoises de la COVID-19, BQC19. So go ahead.
Thank you, guys. Happy to be here. Let me introduce myself a bit more. Because you'll see this has been a very great opportunity for me. I've been at the right place at the right time, and I'd like to convey that we're doing exciting stuff. So I first majored in mathematics a long time ago before deciding to become an infectious diseases doctor. And this research fellowship with Dr. Richards was of course not planned, but then when the pandemic happened, I was sort of at the crossroad between statistical genetics, mathematics, and infectious disease. So this has been a blast for me. I hope I can convey my excitement when I talk about this. Some of the slides you'll see are from Brent Richards from a presentation he did 2-3 weeks ago. They include numbers, because the biobank is still including patients. They include numbers and they're not completely up to date. So just excuse me for this, but otherwise you'll get a pretty good picture of what we've been doing. So I was asked to talk about biobanks, specifically BQC. So we'll start by talking a bit more about biobanks in general and how BQC has been trying to tackle COVID. And then at the end, I was asked to, I think rightly so, to give a specific example of what we actually can do with biobanks. And of course, the focus is going to be on genetics for those two examples, but this is not the only thing you can do with it. And this is really just a superficial view of what you can do with BQC. So let's go in. Biobanks. This is, I'm putting this here because not everyone is aware of the different biobanks that are available worldwide. So what's a biobank? Essentially, there's no specific requirement to call your biobank, your database a biobank, except that maybe it's just a big database. But the first use was in 1996 to refer to the use of human biological repository in cancer research. And of course, most of the early biobanks started as convenient samples. So they just went around in clinical laboratories and just picked up whatever sample they had. They first were centered around diseases. So they were focused on either cancer, or microbiology. So I don't know if you can see my arrow here, but of course the one that really mattered to me for a long time was the ATCC, which essentially defined, allows all bacteriology laboratory to work today. But all of these things, which were standard settings, started essentially as convenient sample disease-centered biobanks. But it worked, it really worked. So Herceptin, I'm not an oncologist, but my understanding is it's still the backbone of many breast cancer therapies, was started as through the data collected in cancer tissue biobanks. In microbiology, as I said, if we didn't have the ATCC, none of clinical bacteriology in the hospital today would work. And then for HIV, for example, that's how they found the agent of AIDS, so HIV. But of course, the scope was a bit limited because it was very disease specific. So you see a shift in focus in the early 2000s from a disease-based biobanks to population-based biobanks. And why that happened there, difficult to say, but I think it was just a few things that happened at the same time. First, we had the internet to communicate on a very large scale. The Human Genome Project kind of proved you can do a very large scale project. And of course, it allowed now for a great analysis and great and greater understanding of the genome. And we had the technology in terms of proteomics, metabolomics, and genomics, and all of that to actually answer questions on a very large scale, because the thing we needed was a large scale. So this is just a small overview of what's going on right now. There are many more biobanks. I tried to focus on the big ones, and I tried to give an international point of view. But we're talking massive stuff, right? The UK Biobank, which started in 2007, 500,000. Danish Biobank, 3 million individual, 3 million individuals have contributed sample. It doesn't mean that they have the genome or any test on all of them, but there's up to 3 million individual. FinGen, China Kaduri, 500,000. The Million Veteran Program, which as the name implies, wants to have 1 million veteran enrolled in their database by 2020. Currently has 750,000. So these are massive projects, and they incorporate different things, as I said, from genomics, but of course, like deep phenotypic or medical history details, proteomics, blood tests on all sorts of stuff, like anything you can ask. Like essentially when they enroll patients, they take a very thorough history, they follow them longitudinally for a few years, and they take a large amount of blood tests, so that if at any point in the future they want to do another analysis or ask another question, they're able to do it by just looking at their biobank, literally, of blood or plasma or serum. So as a researcher, these are maybe things you might want to look into. I'm putting here the 54 gene project, which started last year. I found it interesting. As far as I know, it's the first large-scale biobank in Africa, but it's of course very early on in its infancy. So that's what a biobank is from a population point of view. They're largely exactly the span decade. Okay. So biobanks recap: disease-based, population-based. These are the two big ones. As the name implies, BQC is more of a disease-based, and we'll see why. So March 23rd, I like this picture because I'll ask you guys just crossing his fingers, and he's probably still crossing his fingers nowadays. But yeah, great place to do COVID research, right? So we're definitely the most hit province, and at the Jewish specifically, we were designated as the COVID-19 pandemic hub. So essentially, initially all the cases were transferred to us, and we will be the last hospital with cases most likely. So this was a great place to actually do research on COVID. Going back to biobanks, if you have a population-based biobank, let's say the UK Biobank, and you want to study COVID, so and this is what they did. You know, advantages are it's already established. You already have a large cohort. You already know their medical history. In the case of UK Biobank, you already have genome-wide genotyping or whole exome sequencing available. But it's still just a convenient sample of the population. And of course, I don't want to say that gathering 500,000 individuals in one biobank is convenient. It is not. It's very hard. But the main reason why we could use a UK Biobank is because it was there. That being said, can you really answer questions like that are really finely tuned to COVID-19 as easily with the UK Biobank, for example? Probably not. And this is where the Biobank Quebec, which is one of the many in the world, by the way, this is where Biobank Quebec maybe has a place to shine. So of course, it's more difficult to start than one of the already established population-based biobank, but you can really answer much finer scientific questions on COVID-19. And the questions that you want to ask yourself: as a researcher that's starting up COVID-19 biobank, and these are the questions that we were asking back in March, April, and we're still somewhat figuring them out as we go along. You know, what COVID outcome are you interested in? What biomarkers are you going to measure? What medical history are you going to obtain? What kind of follow-up are you going to do? How are you going to find those patients? Which patients are you going to enroll? Are you going to focus on the younger population, which is when they're really sick with COVID, they're probably more likely to give you a deeper understanding of COVID-19 pathology, but they're very difficult to find because most young patients infected with COVID don't have a significant disease. What's being done elsewhere? Do you want to harmonize your efforts with what's being done elsewhere so that you can gather as much statistical power as you can? Should you align with them? Not everything that's been done elsewhere has been good, so you have to worry. But these are all questions that are very important. I encourage yourself to think about them if you ever want to ask a research question using a biobank, because these are important stuff, and disease specific and population specific biobanks answer these questions completely differently. And so that's important. And of course, how will you fund it? That was less my domain, that was more Brent. So I won't go into too much detail, but I will tell you how we are funded. So I'll try to tell you how we answered most of those questions to try to answer meaningful questions about COVID. So what did we do? So first I'll start with the mission. By the way, maybe I didn't put it anywhere, but all of this stuff, I mean, this is just a summary, but all of this is actually available if you want on the on the BQC19 website. It's all there. Very well explained, open to the public. The mission statement, there's a big PDF about all the protocol. Everything is there if you're interested in it. This is just a broad overview of what BQC19 does, but it's all open science, so you're all open to look at it. But the goal, so enable researchers to better understand determinants of COVID-19 across broad range of disease severity. That's a pretty unspecific goal. So let's try to answer more specific questions. The biobank was set up across the province, so Quebec-wide. Most of the patients were enrolled at the Jewish, as you'll see later on, but this is a Quebec-wide infrastructure, so you can imagine the scale is quite impressive. All right, funding. We were quite lucky. Brent is pretty good at getting funding, but we were also quite lucky to get very good support from the usual funders of science and at the different political levels and at the university. Essentially, we got funded by everyone. But this is important, right? We are parts of other consortium, CanCogen. The NML is the Winnipeg Reference Laboratory. We collaborate with them. COVID-19 Immunosurveillance Task Force, the CIHR, of course. So it was a massive undertaking, which was a bit higher above my head, but it was a massive undertaking at multiple levels to try to coordinate all of this. Governance, I'll skip, but the important part is that there's still, regardless of the coordinating and the fact that a big structure is in place, there's still quite a bit of leeway as to what the different sites can do. So the SHIM was able to run or collect different samples based on their needs or their interests than we were, but there was still a baseline set for everyone, which we'll go into, right? A baseline set of questions and data that everyone had to obtain, which I'll go into the next slide. Or rather, I'll speak about the design. So observational study, multicentric, longitudinal, broad consent, which we'll talk about a bit later, from the patients. So when patients consented to something, we made sure to have them consent to obtain data from quite a large variety of sources and be able to do quite a large variety of blood tests with their samples. Of course, by the way, they could pull consent at any point. Otherwise, that wouldn't work. We ask questions about essentially everything you can think of, and we've gained access to all of their clinical data from the hospital records. And of course, and this is something that was not done in many biobanks, but we felt was very important, we obtained controls that were PCR negative. So didn't have COVID, but either were sick for some other reason, or and COVID was part of the things, was part of the differential, as we say in medicine, of part of the possibilities in terms of diseases, but did not have COVID. So that was very important, and it allows yourself to really ask specific question about what's specific to COVID. In terms of consent, so this is what we had patient consent to. So, and this is all very important and needs to be thought of like when you, like before you actually design your study, right? So consent to recontact the patients, and we have, when we have new questions we want to ask. So for example, in the last couple of months, we've recontacted them to do to look at their antibodies to see if our patients had developed antibodies to COVID. Consent that we'd be able to involve different industry partners. So by industry partners, I mean, of course, pharmaceutical industry, although recently it's really been more Regeneron, which has been very helpful for genome sequencing, for example. So this is not a blank check for the pharmaceutical industry to plow through this, but we want to make this available to all scientists, and so that includes also the industry. Consent for genetics, and that includes whole genome sequencing, that includes whole genome genotyping and GWAS studies, or any future study, or any future technology, so transcriptomics with RNA sequencing. I heard next month you're going to have someone on the HiSeq. I was going to present on HiSeq, so we could, we have consent for this. I don't know if we're going to do it, I don't think so, but we have consent for this. Consent for follow-up, of course, any blood test, any international data access. So consent for their data to be shared across the world. Of course, there's ethical and legal hoops to go through, but consent for this. And of course, open science, meaning that everyone in your, well, I was going to say in the room today, but you're all sitting in different places, but you're all able to actually apply to this data right now. These are the different partners. Where are we? Oh yeah, we're right here. Brent and Jonathan, who's a cardiologist at the Jewish, are leading this here, but we have partners everywhere. Even in Saguenay, I was very surprised, but we have people in Saguenay who are doing something similar than what we're doing across all ages at St. Justin, of course, but they don't have as many severe cases, of course. The MUHC is involved, the SHIM is involved, so really quite impressive. All right, so now let's actually look at what kind of sample and when, what kind of sample we collected and when we collected them. And we had two tracks, essentially, and the colors are important. Try to remember that red is the track for severe cases which were hospitalized, and the yellow is for everyone else. So here's what we did. Again, try to think about this, try to look at this thinking, you know, what kind of questions you can ask yourself, because that's what we were trying to answer. We were trying to be able to ask easy questions, but to also go back to it later on without having to, well, regret not having drawn enough samples. So for the severely ill COVID-19 patients, which for the purposes of this talk will be just the hospitalized patients, okay? There's a spectrum of disease, of course. Essentially, we took a core data set of blood tests, which included quite a few blood tests, which are available on the website. I encourage you to go see. On day 0, and then 1 month later, sorry, X underlined is for PCR positive and negative, and X without the underline is PCR positive only. So for everyone at day 0 and month 1, and for only the PCR positive, all of this. So we're gonna really have a longitudinal view of what's going on with people who have or have COVID or haven't had COVID, depending. So right now our first patients are probably around here, 6 months, but we're set up to really follow them for a long time. And this is what we're gonna do if they weren't severe enough. So quite often, actually, we're able to get some samples on the day of when they were consented, actually, if they were consented inside the hospital, like in the emergency department, but they weren't necessarily admitted. So we would not have necessarily gotten the 2 and 7, but often we got the 0. But this is the plan. So for them, we're going to get out of hospital follow-up blood tests. So again, if we ever want to ask questions on do some other tests on the samples we have, I think we're confident that we have a very, very large biobank to go back to answer those questions. So a small overview of what the actual sample we have up to date, and this is where the next few slides are not up to date. They were probably up to date a month ago, but now with the second peak, we've enrolled quite a few more patients. So the numbers are probably a bit higher than this. Oh, and by the way, this is not just for the Jewish. When there's a difference between the different sites, we'll tell you, but this is across the BQC. Yes. So a bit more than 1,100 hospitalized or admitted to the patients admitted to the emergency. Amongst which about half and half positive or negative. That's a considerable sample. For the ones that were not hospitalized, you can see this here. So, yeah. Now, recruitment. As you can imagine, the JGH being the reference center for COVID, we have quite a few more. The SHIM is following after that. The MUHC, I'm not entirely sure why they only obtained COVID positive patient amongst the hospitalized one, but you can see there's quite a bit of variation, right, among the different sites, but everyone's contributing, and it seems like the infrastructure is slowly getting into place everywhere. Amongst the patients who weren't admitted, again, it makes sense that Saint-Justin has the most, because younger patients, generally speaking, do not need to be admitted for COVID. I guess this is where the MUHC decided to obtain their COVID negative patients amongst the non-admitted. One thing I should say, the SHIM decided to concentrate more on critically ill patients, so patients who went to the intensive care unit, the critical care unit. So that's why there's really not much in terms of patients weren't admitted. They were almost all admitted. Going back to the subject, to the Jewish General Hospital, most patients were consented in emergency, some in the ICU, some in the different wards. We sometimes have had problems with consenting participants, but not being able to obtain blood tests on them. So that's something we should have thought of at the start, a way to find blood tests for them, and we're working on this right now. In terms of the consent, something that's very important if you ever want to plan studies or ever want to plan a biobank, or well, any studies really. Amongst the people who we approached, there's about 14% who refused. I actually was surprised. I was expecting a lot more than that were going to refuse, especially after telling them we were going to sequence their whole genome, which is sometimes a bit shocking to hear, but it seems like people are more and more open to it. 70% roughly of patients actually consented on their own, and 15% we use a surrogate decision maker, so usually a family member. And deferred means that, I mean, by now we may or may not have their consent, but because that was a month ago. But yeah, so in terms of success rate for consent, it was actually not too bad. Now, what kind of data did we actually collect? Everything, really. Demographics. You know, when you read this again, try to ask yourself, what would you have asked for if you had been in our shoes? Because one, we're willing to hear your input, of course, but also it's quite tricky. You want to have good quality data, but also broad data. You want to have data that's easily accessible, but also is still meaningful. So that's what we kind of settled on. So in terms of demographics, address, well, RAMQ and medical record number, this is for internal purposes. We're not going to release that, of course. Date of birth, this is whether they're still alive at any given point. Linkage to other data sets. This is what we meant by well, access to medical records and results from other laboratories. Employment, obstetrical information. Most of our patients, as you can imagine, weren't pregnant given their age, but we were interested initially a lot in the obstetrical and the pregnant population. Habits, smoking, drug use. Try to ask yourself, how would you actually categorize that in a questionnaire? Actually much trickier than you think. It's difficult to actually categorize this, but we did our best, and I encourage you to go on the website to see how. Physical exam. So the vital signs was the most important one, and so for many patients, we have vital signs on enrollment or close to enrollment. In terms of symptoms, we have a big questionnaire about what symptoms they have and since when they've had it. Past medical history from the medical chart or from what the patient is telling us. All of the laboratories in the hospital, at the Jewish at least, and I think at the Shim too, is available. This was a massive undertaking, and we have a big team that I'm gonna thank at the end. We've had access to all of the clinical laboratory results at the Jewish General Hospital. During the admission, we technically have access to all of the medical notes, although it's been very hard to parse, as you can imagine, from the emergency department, from the hospital, and specifically also from the critical care units. We have access, though this has not been finalized yet, to all the reports and images in radiology from all of those patients, which you can imagine is a very large amount of data. Medications, we're still working on that too, but we're working to get all the medications from all the patients that we enrolled, when they were given, how they were given, when they were stopped. So we're going to have a very detailed overview of every patient who was admitted or discharged at the hospital. Interventions in the ICU, that we are working on this. I can't tell too much detail because it's already, because as I said, we're still working on some of the kinks. And then COVID complication, and by that we mean, well, of course, death, but any autoimmune manifestation. So there was a big talk about myocarditis, for example, and COVID-19 is a bit controversial. It probably is a bit flawed, this study, but there was a study about myocarditis and COVID-19. There's been studies about encephalitis and all sorts of other complications. We tried to incorporate that into our questionnaire. The planned analysis we have, they're not all available yet, but we will have them soon. We've sequenced all of the viral genomes, or it will be done very soon. And by we, I should say, actually, it's the LSPQ, so the Provincial Reference Center that's taking care of that part. We've done whole genome sequencing at 30x of almost all participants by now. We've done whole genome genotyping and genome-wide association study, and I put us a little star because we'll go back to this. It's one of the example at the end. Then proteomic profiling with SomaLogic. This is actually being ran right now. So essentially what we did was We sent a sample of blood to SomaLogic, who's going to run measurements using, I think they're using mass spectrometry of over 5,000 proteins. So we're going to have a very deep proteomic profiling of every single one of those patients, which is going to bring up a lot of other questions about how we're going to analyze this. But for now, let's just stick to obtaining data. Transcriptomics, so that's RNA sequencing. Serology analysis is being done, and it's going to be done longitudinally, if I remember correctly. So these are all the things that are going to be available in those patients. So as you can imagine, just a massive undertaking. I wasn't involved in all of this, thank God, but it required quite a bit of coordination. Oh yeah, I mean, the purpose of this slide was mainly to say that in Canada, there's other partners that are doing this, either biobanks or consortium that are interested in this. Internationally, we are part of the International Host Genome COVID-19 Consortium, and I put a star there because we're going to show results from this very soon in the slides. There is one other international COVID human genetic effort that was led by Jean-Laurent Casanova, which you may have heard about. Casanova is out of the Rockefeller, but he has partners across the world. They published 2 science papers about rare genetic variants in COVID-19. So there's other people doing things similar, but those are the 2 biggest international consortium, and the ICDA, the International Host Genome COVID-19 Consortium, is by far the biggest one. So let's go to that example, because before that was a bit dry, and this is more interesting. So what we're doing, what I'm going to present on is what's called genome-wide association studies. You may or may not have heard about this. It's pretty simple, actually. If you go back to your, I'm sure we've all had a crash course in genetics. If you go back to the genome, you remember those base pairs, right? ACTG. At any position, there's a chance that some of us have different variants, so different base pairs. And of course, we inherit one from our mother, one from our father, in most people. In genome-wide association studies, the part that we're interested in are the variants that we've abbreviated as SNPs, so single nucleotide polymorphism. So those are the part of the genome where one base pairs is allowed to change. Okay, so at any one base pairs, it can be switched by another one. So how many SNPs are there in the genome? Well, millions and millions, right? So how many places in your genome can there be a base pair change? Millions. So the idea of a genome-wide association study is at every single one of those SNPs or those base pair variants, you look at whether that base pair change is associated with a specific phenotype. In this case, for example, I've been having COVID-19, severe COVID-19. So that's what we did. And GWAS has been used in many other diseases and yielded considerable insight into the genetic architecture of many diseases, and talking about like anything, cardiovascular diseases, cancers, infectious disease. COVID-19 is actually probably the first large-scale one. Diabetes, osteoporosis, anything that's available in the UK Biobank, essentially. There's been a genome-wide association study on that. So what we do is that every variant of the genome, we fit a linear additive model for the reference allele. So for a long time ago, during the human genome project, we all decided that one of the base pair at every single SNP would be called the reference base pair. It's been done that way to ease calculation. But let's say at a specific SNP, you have a reference that's a G, and the alternative is a T. Okay, so the other base pair that could be that. So one way to fit a linear additive model is if you have a patient with 2T, you give it a 0 at the SNP, G and T, you give it a 1, G and G give it a 2. And then you just simply run a linear regression with this as a covariate on the outcome of choice, which could be, again, COVID-19 hospitalization. Of course, you're going to adjust for many other relevant variables in your model. So you're going to put the SNP and the additive effect, but you're going to put, you know, age, sex. We have to adjust for a thing called population stratification, which just means that the genome is organized differently in different populations, and that can affect your result. And then in the end, what we do, and I love those plots, is Manhattan plots. And we'll show you what it is, but essentially we plot the p-value in a way that looks very much like the skyline in Manhattan with all the different skyscrapers. So as you can see, we're testing millions and millions of variants of different SNPs against the phenotype. And we need a very large sample size to do this. Otherwise, well, we're never going to find something significant. So that's what we did through the ICDA, the COVID-19 host genetics initiative. And then once you do that, you're able to meta-analyze across cohorts internationally. So I'm going to present the example that we did, that we are part of. We're not the only one in it, but that we are part of as part of the ICDA. We're going to present specifically the GWAS on COVID-19 hospitalization phenotype. So again, what we did is for every single SNP, every single variant that's allowed to change by a base pair, we fit that additive effect, accounting for the covariates I talked to you about, to fit this specific outcome, and which we defined as cases as being hospitalized with COVID-19, and the controls being everyone else that's available in the cohort. There is an analysis that we restricted that everyone else which was not hospitalized and with COVID-19, but again, some of the biobanks weren't able to test everyone in their data sets, right, for COVID-19. So to accommodate them, we had a control that was just everyone that's not hospitalized with COVID-19. So looking at that, looking at results from May. So in May, we had those studies. Sorry, I think I cut off something, but on those two columns on the right, the column on the left should be the number of cases, and the column on the wait, what is it? Oh, that's not the correct figure. It should be considerably higher than this. But let's look at, forget the controls. I'm sorry, I took the wrong figure, but the cases were correct. So very few cases back in May. The controls should be, there should be multiple thousand more there. And this is the GWAS that we had. So again, here we have the different chromosomes and the different SNP location. These are the p-values, but because it's minus 10 log 10 of p, the higher a point is, the smaller and therefore more significant, if you will, a p-value is. So it's kind of all over the place. Generally, what we like to see when we look at the GWAS is this nice little peak here. Like if you see multiple variants that are very close to one another and they all kind of are significant, that's suggestive that something's going on here. But here, as you can imagine, because we're doing so many tests, we can't just take 0.05 as the p-value cutoff. So what we have determined back in 2009 to account for different factors is that the actual cutoff for significant test for GWAS should be 5 times 10 to the minus 8. So it should be around here. So we couldn't find anything significant. The line here refers to something else, which I won't go into, but we couldn't find anything significant back in May. Now in June, now the numbers are correct. I'm sorry for the other slides. Now the numbers are correct. So these are the different cohorts: hostage, which is in Italy and Spain, the UK Biobank, GenCovid in Italy, Brazil, Decode is in Iceland. GNH, I forget where it is. Actually, I forget where it is. FinGen in Finland, and BelCovid is in Belgium. All these number of cases of hospitalized PCR positive, COVID PCR positive patients, and controls everyone else in every cohort. back to what I was saying, the UK Biobank wasn't able to test 420,000 patients. So they just assumed that if they hadn't had COVID, well, if they hadn't had a positive COVID test, well, they just had a negative one. Now, in June, what do we see? Oh, now we start to see something, right? So you see chromosome 3, we had nothing last time. Now we have a nice little peak that goes to a p-value of 10 to the minus 15, roughly. And now you see the QQ plot here, which shows that it's probably not just random chance. So already in June, we're able to see that there's a locus in chromosome 3 that is probably determining, in part, your chance of being hospitalized if you have COVID-19. That's quite impressive. That was back in June. Quite a bit of time has gone on. What do we have in October? So October, look at all the studies. So Amsterdam, Decode, as I said, the new one, I'm actually not sure where SPGRX is. There's one from Korea. There's Qatar that contributed. BQC were there now, finally. 23andMe contributed, and 23andMe is a purely commercial, well, you know it, but it's they're in the business of making money from your genome. They contributed data. Million Veteran Program also contributed data. And we have about, I think I'm going to show, no, I'm not going to show, but about 4,000 cases here and more than, I mean, close to 1 million, if not more, controls. That's all across the world. And this is what we find. Now, I want you to look at the scale here. Before that, the scale finished around here, right? 14, 15. Now we're going to p-values of 10 to the minus 70. Chromosome 3, the same locus. So there's something going in chromosome 3. Chromosome 21, which we know is actually this is the chromosome where actually the science papers by Casanova focus their attention. There's a lot of interferon-related and viral immunity-related genes over there. 19, I forget exactly what it is. 9, I forget what it is, but you can see there's something 21 here, which makes sense, and there's a big locus at chromosome 3. QQ plot, again, this is probably not completely random. By the way, chromosome 3, we're still trying to figure out what is so special about it. We have a few hypotheses, but nobody really knows at this point which of the gene in this locus, and there's about 20, 30, is actually responsible for increased COVID-19 severity. But we're working on it. But there's definitely something going on there. So this is the kind of stuff you can do with whole genome genotyping. You can run a genome-wide association study, kind of find out which locus or section of your genomes are important for disease pathology. And then you do further studies to really try and answer questions such as, what kind of drug can I develop to tackle those? And this is what we're going to do in the next one. And one of the things we've done in our lab is using Mendelian randomization to try to prioritize drug development. So we're going to try to go through this. I have 10 minutes. I may fly a bit quickly through this. I apologize. So you found a genetic locus. What now? Can you use this knowledge to find a drug target? And yeah, we can try. So the big question we're trying to ask is, let's say we have a protein level and we have the status of hospitalization in COVID-19. How can you actually say that it's not COVID-19 that's affecting the protein level versus the protein level that you have that's actually making you more at risk? And by protein level, I mean in the blood. That's making you more at risk for COVID-19. And this is very difficult to actually answer as a question. Because of the many confounders and the possibility for reverse causation. So this is where genetics come into play. So if you are aware of genetic variants that make your protein levels vary, okay? Well, those genetic variants are not affected by confounders. Confounders such as social status, confounders such as education, confounders such as smoking, such as if you've been given chemotherapy recently, it's not a affected because your genes are determined at birth, right? Or even before that, actually. So you can use genetic variants to inform protein levels, and therefore, indirectly, forget about this. Therefore, indirectly, you can infer, if you know what's the effect of the genes on the protein level, and you know what the effect of that gene on disease, you can infer this little part here. And forget about pleiotropic pathway, we're not going to talk about it, but that's the whole point. So using GWAS on disease from the COVID-19 initiative, we have markers for disease severity. Using other GWAS published elsewhere, we have markers for protein level. Therefore, just by the neat little mathematical trick, we can actually determine in which direction this is going. And this is essentially what we did. We looked at CISPQTL, which are genetic markers the different phenotypes from the ICDA. Those were the different case control ratios. So hospitalized, 6,000. I said 4,000 before, it's actually 6,000 to 1 million controls. Susceptible, which is just COVID-19 versus positive. So ratio, and very severe is dead or invasive ventilation from COVID versus everyone else in your cohort. And we actually found an interesting gene, protein, I should say. So the OAS protein, which is associated with more and more severe disease if you have a lower amount of it. Well, using Mendelian randomization and genetic studies, we're able to determine that it was actually probably causal for severe COVID-19. OAS is a protein. Interestingly, what it does is it once it sees I'm not gonna go into too much detail, but when there's a virus, an RNA virus detected inside a cell in your body, OAS destroys that RNA. So it's directly virucidal in a way. So it is part of the innate immune antiviral system, innate antiviral immune system, I should say. So we were quite pleasantly surprised to find this. So this is why we do Mendelian randomization. This is why we do genome-wide association studies. This is why we're interested in the genome to find targets for the pharmaceutical industry to maybe develop a drug. So we actually talked to some people in the pharmaceutical industry about OAS, and they're looking at maybe using this to treat patients better. I'm going to skip those because it essentially just said what I said. But yes, summary. Establishing a biobank or using its result require careful consideration and a lot of planning. Okay, a lot of planning. And even if you only use the results from this, you need to be aware of the different shortcomings at every single step of the design of the biobank you're going to use. Using a biobank like the UK Biobank to answer questions will only allow you to answer specific types of questions, whereas PQC19 will allow you to answer other questions. So you need to be able to have the design and the planning that went into the biobank you're going to use. BQC is the largest biobank in the country. It's amongst one of the largest COVID-19 biobanks in the world, not population-based biobanks. And then one of the goals of all of this eventually, and I want to emphasize this, is really to find drug targets through the use of GWAS, proteomics, and all of the other analysis I spoke about. And open science. So if you guys are interested in this, the whole genome genotyping for every single patient is available. You just have to apply for it on the website. It's really not that complicated. I think about 15 groups have already accessed it. I'm not sure where they are. But if you are interested, this is something we can set up. I think that's, yeah, those are my last two slides. If you guys want to ask questions, go ahead.
Great. Thank you for the amazing presentation, Guillaume.
Thank you.
I noticed that a lot of you guys showed up after we started the presentation. So there are 30 of you now. I think I started when we were around 7.
Oh, okay.
So for everyone who showed up late, we will have a recording of this available on the EMGSS website, emgss.org, in due time. So I'd like to open the floor for questions if anyone has any. You can raise your hand or just unmute yourself.
Can I ask a question? I'm Pierre Paradis, the Davis Institute with Ernesto Schifrin. In fact, I have 2 questions. What was the percentage of lab tests done on the COVID patient at the admission?
I wouldn't be able to tell you exactly, but I can tell you that every single so it's a bit tricky because we have access to all the blood tests from the clinical laboratories. So in that sense, essentially, if you showed up to the emergency department at the Jewish and you were eligible to be enrolled and you gave consent, we have access to the blood tests that were done that day. There was a more standardized set of blood tests that I know that 100% of the patients who were hospitalized got that standardized set of blood tests. But for those who only were in and out of the hospital, let's say because they went to the emergency department, we may not have it. And I don't have the exact number. But what that means is that more eyeballing it, we probably have that standardized set of blood tests on more than 70% of patients, but we have the common stuff like CBC, liver profile, coagulation, Chem7. On many patients, we have procalcitonin, and some like 200, we have interleukin 6, for example. Like we have this on almost everyone.
Okay. And what about the onset of symptoms? Do we have this information?
Yeah, that's a very good question, actually. It is part of the questionnaire. I forget to check how many were actually filled, but for the most of them, it was filled.
Okay. Can I ask another question?
Absolutely.
The last one. So why the data bank contains so many COVID-19 negative patients?
So yeah, that was a design choice that we made. First of all, I mean, the easiest question you can ask yourself is there something that makes you susceptible to having COVID or not? And the only way you can really be sure is to enroll patients who don't. There was also a question about, for example, if you have pneumonia because of Streptococcus pneumonia, one of the most common bacterial pneumonia, and you end up in the ICU. A question you can ask yourself, because you're very sick, question you can ask yourself is there fundamentally a different pathophysiology to you being in the ICU because of pneumococcal pneumonia versus COVID-19 pneumonia? And so to do this, you need controls that aren't COVID positive.
Okay, thank you.
But it's a good question because to do this, it requires more design. And more, like you need to think about it a bit more. And it's not everyone in the world that did this. Many other COVID-19 oriented biobanks in the world really only enrolled COVID-19 positive patients.
Okay. Thank you.
Were there other questions? I had one for myself, so I'll just add that in. It was interesting to see that you record medical notes and interventions from each patient. I was just wondering, is someone actively transcribing the data from each doctor's notes, or how is that standardized?
So that's a very good question. So no, you have to do, like, for example, for radiology report, we're looking at actually uploading or downloading, I should say, all of the free text report from the radiologist. We take the report and that's what we're going to make available. For things that are handwritten, as you can imagine, it's quite a bit more painful. So we had to have a questionnaire, well, like a database that we enrolled an army of scribes that essentially went through the charts, and it's true that this, I mean, you have to balance two things, right? You have to balance precision, but also with like what's feasible. And yes, if you have a preset list of questions that you want to look at into the medical records and then put into your database, that probably makes it easier to do on a larger scale, especially because it's all done by hand. But you're going to lose some finer detail. There's no question. So yeah, we have an army of scribes that, are they here? They should be here. Yeah, exactly. So some of them are here, actually, and the clinical research team. They're not all the scribes, of course, but quite a few, like Zaman helped quite a bit, Myriam, Nardin, they helped quite a bit to extract data manually from the charts.
And one final question from me. I noticed the Saguenay-Chicoutimi group was quite small in comparison to the Montreal group. I know that, I mean, there are less cases in rural locations, but I'm wondering if you guys are looking into other rural locations, seeing as it might be a different representative population.
Yeah, that's a very good point. My rumors have it, but I haven't checked the numbers, that now the second wave is hitting different parts of the province a bit more equally, and they may actually be enrolling more patients. So this may actually change. In terms of other sites, I mean, we're always looking for more, but the thing is, for things like this, you really need you know, a champion at your site that's going to be the one that's going to carry this project through. Here it was Brent and Jonathan who did a great job, and this is not a judgment on rural sites, but like, they may not have either the infrastructure, the interest, of the manpower to do it. So this was the site who were willing to go with us. This is not because we excluded other places, but this is maybe because they were lucky enough to have to be able to do it. I try to stay politically correct because I understand it's hard to set up this in the time of a pandemic. Right? Like it's easier said than done.
So any other questions before?
Yeah, there's someone who sent me something on the chat privately. So I'm just going to read it. Okay, so they just wanted clarification as to what PCR positive and negative meant. That's a good question. I'm sorry if I didn't go through it. PCR is, it stands for polymerase chain reaction. It's a technique used in clinical microbiology and research quite a bit. It's a specific type of nucleic acid amplification test. And essentially, in the context of COVID, the way we've been using PCR tests is in clinical microbiology laboratory, so in the hospital laboratory for diagnostic purposes. So those are the tests that you see on the news or on TV where they stick something up your nose and it's very unpleasant, and they send that to the laboratory, and then they use that PCR technique to see if they can find SARS-CoV-2, which is the agent of COVID-19. So that's what I meant. If you were PCR positive, it means that the actual swab they did was positive, so they could find the virus. And if it was negative, they could not find the virus. The person who asked me this, I'm not going to say the name because it was privately. So the person who asked me this also asked me if there was any other additional tests for the diagnosis of COVID-19. I mean, yes, there has been other tests that that have been looked into that are not really included in BQC19 because they've never really panned out in clinic. But those include antigenic tests, which essentially look at the virus in a different way using antigen that the virus is producing. It's not as sensitive, so you're going to miss quite a bit of cases. And of course, there's also antibodies, but there's always a delay between the infection and the antibody response. So you're also going to miss a lot of cases. So really, in life, the only tests we have are nucleic acid amplification tests based for the diagnosis of COVID-19, and the most common one is a PCR.
I have a quick question.
Yes.
Thank you for your talk. I'm really sorry I missed this information. How many patients are involved in the full cohort, and how many are positive for COVID-19?
Oh yeah. I'm going to show you the slide.
Thank you.
Those are, I mean, those are the people who are hospitalized, and then the next one is people not hospitalized. Again, with the little caveat that this was about a month ago. So at the start of the second wave, and I know we've enrolled quite a few more.
Okay, because we have received the first set of data and it's lower than that, so that's why I'm asking. And
so did you ask for the data at the JGH? Because this is from the whole Biobank Quebec.
Okay.
The other groups weren't. I mean, I'll skip you the politics part, but we were a bit more. We were able to have a better infrastructure to set up the sharing part of the biobank. So the Jewish was able to share data before others. So that's why there might be a discrepancy.
Okay, okay, I understand now. And I have another question. Did you measure the viremia in patient at hospitalization? I mean, whatever. I mean.
We have the capacity to measure it, and I know the SHIM is doing it.
Okay.
But viremia is tricky. I'm not sure how much you know about those nucleic acid amplification testing, but you need some sort of standard to quantitate the amount of virus, right? So it's very tricky. And for now, we don't, in clinical medicine, we don't have a quantitative method. We have the semi-quantitative that's based on cycle threshold, but to do the jump from cycle threshold to quantitative, like full quantitative, requires quite a bit more thought, and nobody's done it at a clinical level. So I can tell you that at the JGH we haven't done it, but we have the samples that we can go back to if in case at some point we could do it. At the SHIM, they've started to do it, not for clinical reasons, for the biobank. So only research.
Okay.
All right. Well, I think that's everything for now. If you have any questions, you can put them through me to Guillaume through my email, which is on emgss.org, or you could potentially also find it. I think Guillaume's email is on the Richards Lab.
Yeah, pretty easy to find.
So for anyone who's interested in the November seminar, I'll send you the link just now. And I want to thank Guillaume again for a great seminar. I think everyone came by was interested, and it was a very useful talk. So yeah.
Thank you guys for inviting me.
Thanks everyone.